everyone, and welcome to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. One of the things you may notice up front with this slide is we weren't able to have Dr. Chin join us, but fortunately we were able to get Florian join us today. So really looking forward to that talk. If you are experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you're having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all of the associated materials within a few days. If you have questions during today's webinar, let us know about those using the GoToWebinar chat box. Today's talks will each be 20 minutes, followed by 10 minutes for questions. Benjamin Schlesinger, who is an ARC Future Fellow and Independent Group Leader at Australian National University, is with us today to moderate your questions and read them aloud to our panelists. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. This webinar is part of our new virtual research talk series that we created in response to the closure of most universities due to the COVID-19 pandemic. Please visit our webpage for more information on this series and to sign up if you'd like to be considered as a speaker. You can find us on social media at plantae_org and at ASPB. For those of you listening to this as a recording, feel free to reach out on Twitter with follow-up questions or comments using hashtag PlantaePresents or post those on our blog. Thanks again for joining us today. I'm going to turn it over to Benjamin now to introduce our speakers and moderate today's session.
Thank you very much, Katie. Welcome, everybody. Before we get into it, just wanted to say thanks to ASPB and especially Mary, Katie, and also Jürgen for co-organizing this. This will be today my last moderation before the break, before the ASPB Plant Biology Conference break you hopefully all go to. I hope you enjoyed this. I enjoyed this a lot, and I really would like to say thanks to the Society to support such a forward-looking approach, and we hope we will continue this in future. All right. I'm really excited about the two talks today. So it's Florian Busch and Katie Murphy. Florian will take us away initially. Thanks, Florian, for jumping in the last minute, probably like 20 hours ago. So really appreciate this. So Florian Busch is a research fellow at the Australian National University. He obtained his PhD from the Heinrich Heine University in Düsseldorf, Germany, during which time he worked closely with Norman Hühner at Western University, London, Canada. After several years of postdoc work with Rowan Sage at the University of Toronto, Canada, and Suzanne Cameron at the Australian National University, he moved on to his current position with Graham Crawford. He will start his own group at the School of Bioscience at the University of Birmingham, congratulations, later this year. Florian is a theoretical and experimental plant physiologist interested in all aspects of photosynthesis. His focus is on linking different photosynthetic processes to mathematical models to study the biochemical limitations of carbon fixation and to gain a quantitative understanding of how plant carbon uptake responds to change in the environment. I'm looking forward to the talk. Thank you very much, Florian, for jumping in last minute, and please take it away.
Okay, well, before I start, I'd like to thank Benjamin and Mary and Katie for giving me this opportunity. Even though it was short notice, I very much appreciate this. Yeah, for those who don't know me, I'm a plant physiologist interested in photosynthesis. And in particular, I'm interested in how different aspects that influence plant carbon uptake interact with each other to determine how much carbon the plant can actually assimilate, and how all these photosynthetic processes respond to the environment and changes in environmental conditions just so that we have a better understanding of photosynthetic carbon uptake and how that might change with environmental changes. So two of the main drivers of photosynthesis are obviously CO2 as the carbon source that for the assimilated carbon, and light that provides the energy to fix this carbon. And while this light energy is actually ending up in the place where it's needed right away, it's absorbed by chlorophyll in the chloroplast, the CO2 has to first diffuse from the atmosphere into the chloroplast where it's needed. And I have this little schematic here to demonstrate this. So we have the CO2 in the atmosphere that needs to diffuse through the stomata into the intercellular space, and then onwards through the cell wall and the membranes and so on into the chloroplast, where it's ultimately assimilated by rubisco. And these the diffusion pathways and stomata and membranes and so on along this diffusion pathway provide a resistance for CO2 diffusion. So we call this stomatal resistance, RS, or mesophyll resistance, RM, that sort of cause a drop in CO2 gradient along this diffusive pathway into the chloroplast. So the chloroplastic CO2 concentration is actually quite, in general, much lower than what we have on the outside in the air. So rubisco is an enzyme that doesn't just use CO2 as a substrate. It can also use oxygen as a substrate. And the product resulting from that is poisonous to the plant, and the plant needs to metabolize this. And it does that via the phororespiratory pathway involving peroxisome and mitochondria, and in the process of which some CO2 that's previously been fixed by the plant is released as, or some carbon that's being fixed is released as CO2. So we have this balance, that the net assimilation by the plant is sort of the carboxylation rate, so how much carbon rubisco fixes, minus the CO2 that's released from photorespiration and respiration. And all this ends up making up how much plant the carbon actually can take up. And so it turns out that the amount of carbon being taken up by rubisco, or CO2 being fixed, versus the amount of oxygen fixed, is very much dependent on the CO2 concentration inside the chloroplast. So this really determines the amount of carbon that the plant can fix. But how do we know what the CO2 concentration in the chloroplast is? Well, there's a fairly simple, straightforward way to measure the intracellular CO2 concentration. But getting at the chloroplastic CO2 concentration is much more difficult. And luckily, there's this technique that we can use, which is based on carbon isotopes. So the CO2 in the atmosphere comes in different flavors, if you will, with a lighter carbon or with a heavier carbon. And the lighter carbon has the benefit, or has the property, that it actually travels faster in air. So the diffusion, or in water, and the diffusion, therefore, is much faster for the lighter C12 CO2 than it is for the C13 CO2. And for the same reason, or similar reasons, the lighter CO2 also reacts faster in biochemical processes. So rubisco prefers to take up the light C12 CO2 over the C13 CO2. And that, in sum, that means the plant preferentially takes up the light C12 CO2. And we can measure that. And we can also then use this property to deduce diffusion or biochemical functions inside the plant. And Farquhar et al. Almost 40 years ago or so came up with this equation here that makes use of a measured carbon isotope discrimination value by the plant. So that's essentially a value how much the plant prefers the light carbon over the heavy carbon, and attributed this to different diffusional or biochemical properties inside the plant. So the first term here is describing the fractionation that happens with the diffusion through the stomata. The second term here is related to the diffusion through the mesophyll, and then we've got a term here that's related to the biochemistry of Rubisco, and two terms here related to the fractionation imposed by respiration and photorespiration. So, and then the idea is that we know all these parameters in this equation, or at least we can measure them, or at least we have a good idea about them, except for this one here, the CC, the chloroplastic CO2 concentration. And from that, we can then calculate what the drop is in CO2 concentration from the intercellular to the chloroplastic concentration. So that's sort of the principle behind it. So historically, or traditionally, we've assumed that the CO2 coming from the mitochondria magically ends up in the chloroplast. This, I guess, was done because of simplicity. It's much easier to describe this, and maybe because we couldn't determine it any better anyway. But a few years ago, what Tolan et al. Pointed out is that really the CO2 is coming out of the mitochondria and ends up first in the cytosol. And the consequence of that is that the flux of CO2 from the cytosol to the chloroplast is really the sum of the flux of CO2 coming in from the intercellular space and the CO2 coming in from the mitochondria. So we have a different flux across this part than we have across this part of the mesophyll diffusive pathway. And as a consequence of that, really what we have to do is we can't consider this as one single resistance, but we have to split it up into two components. So what we call here the wall and plasma membrane component and the chloroplast component. And this has a very profound effect, as I will outline in a second. So here I'm plotting GM, which is mesophyll conductance, which really is the inverse of this resistance. So it describes how easy CO2 can diffuse into the chloroplast relative to the CO2 concentration. So if we have a single resistance, all things equal, we should assume that the GM value, the mesophyll conductance value, is constant. If we have this two resistance situation where the CO2 from photorespiration comes in to the cytosol, then the apparent GM that we observe will depend on how much CO2 comes from photorespiration. So we should expect GM to change with both CO2 concentration and oxygen concentration, which are both affecting how much CO2 is photorespired inside the plant. And as you can see here, as you go to lower and lower CO2 concentrations, GM goes to lower and lower values and reaches 0 at some point. And conductance of very low means it's very, very hard for CO2 to actually diffuse into the cell. So at this point here, it's physically impossible to get CO2 into the cell. And if you compare that to some constant GM value, you can appreciate that this is a big difference, and it makes a difference in how we view what's going on inside the plant. So a few years ago, when I started looking into this, I was like, well, now that our measurement techniques are getting better and we can actually measure GM at maybe lower CO2 concentrations, can't we just use this isotope isotope technique and see how GM varies with CO2 and oxygen, and see if you fall onto this line or if you fall onto the green line up here. So when I did this, this is sort of what you come out with, and it looks like it doesn't really fall on either one of those lines. But there's an additional problem. So these fractionation factors here, B, E, and F, we sort of have an idea what they are, but we're not very clear. And the values for E and F, for example, they can vary by, let's say, 100% or more of what people have used in the literature. And it turns out if we just vary those a little bit within the range of what people have used, we can get the estimated GM to be flat and insensitive to oxygen. Or we can even make them turn the other way around. And I don't know what happened just here. There we go. And make GM increase as we go to lower CO2 concentration. So that shows us that this method here is, in this case, is not really that useful to get us a good value of GM until we actually know exactly what these fractionation factors are. So together with a fellow postdoc in the lab, Misha Holloway-Phillips, we devised a method how we can actually test this model itself independent of these fractionation factors and see how it performs. And the idea was that we measure GM with different gases, different CO2 isotopic compositions. And given that GM shouldn't change when we change the measurement gas, we would expect that GM estimated with both these gases come out with the same values of GM. But as it turns out, if you use these two isotope composition of the measurement gas, the GM that we estimate deviates between these two gases, the lower the CO2 concentration. And we also end up quite often with negative GM values that are not really meaningful. So negative conductance is, you need to come up with some way to explain this. So this is not really that meaningful, which means the model itself has some issue because the deviation is independent of what B and E and F we use for that. It also points us towards what the issue might be, because the only way, the only part where the isotopic composition of the measurement gas goes into the equation is in this respiratory term up here. It's built into this E term. So in order to track down and see what this model is doing and how we might be able to fix it, let's dig a bit deeper into the basis of carbon isotope discrimination. So Farquhar et al. Have made this really simple assumption that the isotope ratio of the assimilates is equal to the isotope ratio of the carbon that's being assimilated. So that's the chloroplastic CO2 concentration and the fractionation by Rubisco, minus the ratio of the carbon flux coming off from photorespiration and from respiration. And maybe a bit of an easier way to look at that is with this schematic here, where you have the carbon being assimilated by Rubisco and fractionated upon by Rubisco, fills up a pool of assimilates from which respiration draws off some carbon and photorespiration draws off some carbon. So the important bit here, though, is that if respiration preferentially releases, say, light carbon, that means heavy carbon is left behind and making this pool of assimilates heavier. And that in turn means that the source carbon for photorespiration to draw upon is also heavier. So if light carbon is respired here, the carbon coming up from photorespiration becomes heavier. So now the question is this actually what's going to happen in the plant? And to assess that, let's talk you through this schematic of CO2 assimilation here a little bit. So in the center, we have Rubisco that uses the CO2 in the chloroplast and fixes that to produce phosphoglycerate. Or alternatively, it uses oxygen and produces phosphoglycolate. And that phosphoglycolate is the toxic compound that needs to be recycled in the photorespiratory pathway, where the CO2 is released from photorespiration. And then all the PGA produced in both these processes is then recycled in the Calvin-Benson cycle back to REBP, which is the other substrate for Rubisco. And the fixed carbon that's left over is essentially what makes this assimilatory pool from which respiration draws upon. So, as I previously mentioned, if this situation is true and the heavy carbon left behind from respiration affects photorespiration, that is equivalent with having some carbon flux from this assimilatory pool back into the Calvin-Benson cycle that can then make its way around into photorespiration. And the question is, well, can we assume this is a good assumption, or do we have to revise that? So let's just look at sort of the most simple approach to respiration, where we have pyruvate being decarboxylated in the TCA cycle and release as CO2. So pyruvate can be decarboxylated and produces energy, and that's often how we view it, as respiration as the source of energy production. But there might be two issues with this. For one, if we take all the pyruvate and decarboxylate it, that means the entire pool of pyruvate carbon ends up, or respiratory pyruvate carbon ends up as CO2, which means the CO2 has the sum of those CO2s have the same isotopic composition as the original pyruvate from where the CO2 derives. So that is equivalent with saying there's no respiratory fractionation happening. And there's evidence that respiration actually does fractionate to some degree. The second point I want to make is that many of the metabolites in the TCA cycle are actually used by the plant for other things. So acetyl-CoA is used for fatty acid biosynthesis, and other compounds are getting used for amino acids and nucleic acids. And these are compounds that actually stay in the plant and make up plant biomass. So there's carbon left behind here that is not respired and actually stays in the plant. So if you come back to our graph on the left here, this is equivalent with moving this assimilatory carbon pool out of here. So if you say this is the carbon that actually makes up the plant, the biomass of the plant, then we can say part of that carbon is carbohydrates, which come from the Calvin-Benson cycle. Another part of the carbon is lipids and proteins and so on, which also come from here, but which have respiration involved to become these compounds. And the other assumption then is that this is not making its way back into the Calvin-Benson cycle. So if we then go ahead and derive an isotope discrimination model that takes all this into account, we end up with this equation here. So this is an equation that considers RD to be isotopically disconnected from the Calvin-Benson cycle. And I don't blame you if you say this looks like the old equation that Farquhar et al. Came up before, with one important difference. So Farquhar et al. Had respiration scaled with the carboxylation rate, whereas this new model here has respiration scaled with the assimilation rate. So as we go to an assimilation rate of 0, this becomes very large and goes towards infinite. Whereas in the old model, this stays quite small. And infinite has obviously a very big effect and has also a very big effect on the mesophyll conductance we will calculate from that. So if we take this new model and we measure our assimilation rate with C2 going to smaller and smaller values, we end up with the assimilation rate going towards 0, and thereby having a big effect on this term here. And when we calculate GM from that, we can see that the GM we calculate now from that is independent of the gas tank that we use, which is really what we're looking forward to, because we don't want our measurement to actually affect the value that we measure. So from that, we can say that this new model actually much better describes GM, and we can be much more confident in estimating these GM values. So we still don't really know what those values B, E, and F are, and they still might vary, and we might be using different values. But what happens if we use different values? Let's say if we vary this value F within those parameters or within the values that people are using, pretty much for any value that we use with the old model, we get this big deviation going to low CO2 concentrations. Whereas with the new model, independent of our value F, we get a close agreement between our two measurement gases. And similarly, if we vary E, the other parameter that's not quite well known, we get a close agreement with the new model, but not with the old model. And maybe a second thing to notice, so depending on what value of E and B and E we use, unless we know the exact value, we still can't get an actual correct quantitative estimate of GM. But what we can do is, whatever value we plug in, we see this curvature going to 0 GM towards the compensation point, independent of those E and F values. And from that, we can conclude, well, it follows this purple line, which corresponds to this two resistance situation, rather than this constant mesophyll conductance, which would correspond to the single resistance model. So from that, we can conclude this simple view of where the CO2 is being released is sort of too simple, and we actually will have to move on and think about incorporating this two resistance case into how we view CO2 diffusion. And ultimately, this will help us assessing the CO2 concentration inside the chloroplast, how easy it is for CO2 to actually diffuse into the chloroplast, and what consequences it has on photosynthesis. And with that, that concludes my talk, and just leaves me to thank my partners in crime on this work, which is Misha Holloway Phillips, who's now a postdoc in Bern, in Basel, as well as Graham Farquhar and Hilary Stewart-Williams, who were very instrumental in setting up the machine and thinking this through. And yeah, thank you all for listening and having me.
Thank you, Florian. Thank you very much. So we have a little bit of time for a couple of questions. Let me pose this up for you. So the first one was, based on the overall model, which part of photosynthesis would you target first to increase carbon assimilation and therefore plant yield?
Oh, that's the holy grail question. So one of the things I'm really interested in is what actually limits CO2 uptake under any given condition. And it turns out that there's different processes that are limiting CO2 uptake under different environmental conditions. And they change very quickly depending on things like light availability and or temperature, or they can change long-term based on water availability or climate the plant grows in. And each of those limitations would require a different approach to remove that limitation. So if Rubisco turns out to be the enzyme that limits CO2 uptake, then well, you might be able to fiddle around with that and remove that limitation. But if it's light availability and you have plenty of Rubisco around, then it's not going to help. So it would have to be targeted to a specific condition, or maybe a specific average condition where you want to grow your plant in. And then you'd have to look at what is actually limiting assimilation rate under those conditions, and then come to a conclusion of how to remove that limitation. But I don't think there's a one size fits all.
Not that easy, I guess. All right, so in the literature, I think photorespiration is often seen as a wasteful process. In a lot of like GM or approaches right now, try to reduce photorespiration or completely get rid of it. Do you think that's a good thing?
So I don't see photorespiration as a wasteful thing in general. So yes, there can be too much of a good thing, and photorespiration can be wasteful to some degree. But just as respiratory processes feed into producing metabolites for other purposes, so does photorespiration. So the glycine and serine that's produced via the photorespiratory pathway is used for protein synthesis, for example, and required by the plant. And in fact, photorespiration is the primary pathway to produce these metabolites. So if you reduce photorespiration too much, you sort of squeeze out other metabolic processes. So again, it depends on the fluxes and the magnitude of those fluxes, whether there's too much and you can reduce photorespiration a little bit, or whether photorespiration is actually doing a good thing. And it turns out the carbon that ends up in photorespiratory glycine and serine can actually increase the carbon uptake of the plant altogether. So it can actually be a good thing, not just neutral, but actually a positive thing.
All right, maybe we have time for two questions, I think, depends how long your answers are. So there was another question, whether we can increase assimilation of CO2 through CAM plants.
I would say, again, depends on the condition you want to grow your plant in. CAM is very good to grow under low water availability. So they can have very big benefits and produce a lot of biomass under those conditions. But CAM has a carbon concentrating mechanism that costs energy to begin with. So in a condition where this energy is not well invested, you might be better off with a C3 plant. So depends.
There are a couple more questions, but we will pass them on to you later. So we're going to take the last one now. So you came up with a nice new model which fits the data better, to my understanding. So how does your new model help people who want to improve climate models, for example, to predict CO2 uptake in forests or ecosystems? Does this have any impact on these calculations?
I would say so, yes. So anything, I mean, ultimately it improves our estimates of assimilation rate under different conditions. And under some conditions, the difference between these two models might be small, and under other conditions, it might be large. So if anything that depends on a good prediction of A, I think would benefit from improving that prediction. And I think that's what our new model does, is improves our estimate of A when we model it from environmental conditions.
Well, thank you very much, and thank you very much for jumping into this last minute. I think it was exciting to have a talk which is like hardcore plant physiology. It is really nice, and carbon is such an important molecule, and carbon fixation too. So thank you for the talk. So we will move on to the second talk. Thank you, Florian. To the second talk, which is from Katie Murphy. So Katie Murphy is from University of California, Davis, a lovely place where one of my alma maters. So exciting to have a talk from UC Davis today. So Katie is a PhD student candidate into the therapy lab at the University of California, Davis. Her research focuses on terpenoid biosynthesis in maize and how these molecules relate to the plant stress response. She holds a BS in chemistry from Stanford University, where she studied answer development in maize with Dr. Virginia Walbot. Katie is the early career representative to the ASBB Women in Plant Biology Committee. Is an ASBB ambassador. She recently won the UC Grad Slam for the best short presentation of her research. You can also find her on Twitter at GMO my God. Right, I didn't know I get this right. Thank you very much, Katie. So Katie will talk about today with terpenoids in maize from biosynthesis to bioactivity. It's great to have you. Also, great to see all the service to society. Very much appreciated. Please take it away.
Thanks so much, Benjamin, and a huge thank you to Benjamin, Katie Rogers, Mary Williams, and everyone who's worked so hard to put these seminars together. I'm glad I can see all of you virtually and be here today. So to everyone watching live and watching the recording, good morning, good afternoon, and good evening, depending on where you are. I'm glad we could all come together today. And I want to start off, like he said, I really love ASPB and our society, and I want to share a few of the things that I'm involved in so that you all can get involved as well. So the first is the Women in Plant Biology Committee. We're a committee that's working on providing resources and a voice for women in our society. So if you want to get involved, if you want to volunteer, you can find us on Plantae and on Twitter, or you can ask me any questions that you have. We'd love to have you participate in our community. The next is the brand new early career section of ASPB, which I'm excited to be a part of. So this is to help advance early careers, provide an arena to network and gain other opportunities for grad students, postdocs, or anyone early on in their careers in industry or academia. It's only $5 additional to your ASPB membership. You can find it online and ask me any questions. So now let's get into the star of the show, my two favorite things, terpenes and maize. Today we're going to do a brief overview of diterpenoids. These are the molecules that we study in my lab. We're then going to get into biosynthetic pathway discovery. That's what we typically do, and I'm going to use these new molecules in maize as an example of how we do that. Next, we're going to get into what these molecules do in maize, and here I'm going to give you some appetizers on biotic stress response, abiotic stress response, and it's probably going to be like an appetizer. It's going to taste good, but will leave you unsatisfied and wanting a full meal, and that's the goal because it's keeping it short today, and if you have more questions or want to read more, I can give you some resources, and we can talk in detail later. So let's dig in now. What are diterpenoids? I think of these as medicines for both plants and people. All diterpenoids come from one central precursor, that's GGPP, the 20 carbon compound you see on your left. A series of enzymes then convert this single molecule into a large diversity of structures, the most famous of which are probably the gibberellins. These are essential plant growth hormones, but are also important as plant defense metabolites, also called secondary or specialized metabolites. Now, humans, we've gotten really good at exploiting plants for our own uses, so plant diterpenoids also play an important role in our own lives. Two of the most famous diterpenoids are Taxol, which is a chemotherapeutic compound that comes from the Pacific yew tree, and artemisinin, which is currently the best treatment for malaria. So terpenoids are present in our everyday lives, even if you don't think of them as often as we do. So how do you make a diterpenoid? Again, here on your left, you start with GGPP, this one central precursor. Then you have a series of enzymes. You start with a class 2 diterpene synthase, followed by class 1 diterpene synthase, which generates that carbon scaffold. And then cytochrome P450s decorate the scaffold, typically with oxygen functional groups, in order to provide a variety of functions and additional molecules. So total, we know so far of over 10,000 known diterpenoid metabolites in nature. This is huge. That's a huge variety of chemical structures. We also know that they're typically species specific, and different plant species will have different blends of these diterpenoids. So people often ask me, I don't understand, how do you get 10,000 metabolites, but you're telling me one precursor? So I like to think of it in terms of ice cream, which I'm going to share with you all today, and also because I like ice cream. So we're going to think of the components of our ice cream cone as the different molecules along these pathways. Here we're starting with a single precursor, and if I give you only one flavor, you only have one resulting product. Now, if I change it up and give you 3 flavors of ice cream, you can now make 3 different ice cream cones. Here, this is an analogy for how subsequent rounds of gene duplication and neofunctionalization through small changes in the active site can lead to different molecules, or here, different flavors of ice cream. Now let's take it a step further. As Emerald says, kick it up a notch, and I'm going to give you toppings. Now, assuming that you can just put one topping on each ice cream, you can now have 15 different combinations. Here, we're understanding substrate promiscuity. Each topping can go on every single different type of flavor of ice cream. So we know that enzymes can take multiple substrates and also generate multiple products. Here, it's not just a pathway, it's an interconnected biosynthetic network that leads to a huge amount of diversity and what we consider a large chemical space. So if we overlay the diterpenoid biosynthetic pathway on this analogy, the result is species-specific blends and this huge number, over 10,000 metabolites. Again, that's just so far. So now let's dig in and let's get into my favorite plant, which is maize, and which is also the economically most important crop in the United States. What did we know when we started this project? Well, we started looking through the genome because fortunately we have an excellent resource, and we looked for genes that were homologous to known diterpene synthases. Now, fortunately, we say what keeps us in business is knowing that you found a diterpene synthase doesn't tell you its function. Again, small changes in the active site, which is really useful for neofunctionalization, means that you really have to test each enzyme to know what chemical structure it's making. So when we looked through Maize for homology, we found 4 class 2 diterpene synthases and 5 class 1 diterpene synthases. You can see them here annotated on the 10 chromosomes of Maize, and they were spread out. Now, some specialized metabolite pathways are clustered, which means the genes are physically located near each other on chromosomes. This is also true of diterpenoids in rice. But in maize, this wasn't the case, so there was no clear indication to us right away which genes might be working together in pathways. Now, I'm going to show you what the biosynthetic network in maize looked like when we began this project. So on the left, you have the gibberellins, these essential growth hormones, and a lot of great work went into elucidating those pathways. We also had the coralexins. This is a group of metabolites that were known to play a role in the plant stress response, something we're going to get into later, and they were known to originate from AN2, ZMAN2, but the rest of the genes in the pathway weren't understood. So here we were taking a metabolite to gene approach to try to understand which genes are involved in this pathway, now that we had found some through homology modeling. So this is the method that our lab takes, which we like to call rapid gene characterization via co-expression assays. So we start with transcriptome profiling. We've chosen these genes by homology modeling, and we're now looking for which of these are expressed at the same time, in the same tissue, and for us under the same stress conditions. Again, these coralexins were strongly implicated in the plant stress response. So here you can see that AN2, KSL2, and KSL4 were co-expressed together. So this told us these are the first ones that we wanted to test. We next clone these genes, or nowadays we often synthesize them, into multi-gene plasmids. In this way, we can mix and match the different plasmids to make up different biosynthetic pathways that might make up a network. We then take these plasmids and we transform them into a heterologous system, E. coli, which is my personal favorite, and remember that for later on. And occasionally we'll use yeast or tobacco, Nicotiana benthamiana, using agrobacterium infiltration. So now that we have a heterologous system that has taken in these genes and producing our molecules, we have to figure out what they are. So here we use GCMS, that's gas chromatography, mass spectrometry, occasionally LCMS, liquid chromatography. And if we have a new molecule, one who we don't have a standard for, or we can't find in our mass spectral library, we then perform NMR to characterize that structure and its specific stereochemistry. So again, let's look back at where we were when we started this process, and now I'm going to show you where we are today with the maize diterpene biosynthetic network. Now, I realize this is a little crazy. We're going to break it down, but I just wanted to show you the progress that we've made so far, and we're really excited about these new branches and new areas to explore. So to start, let's look with just the class 2 diterpene synthases. We already knew about AN1 and AN2, both of which make Ent-CPP, and now we've added on CPS3 and CPS4. You can think of this as I've given you 2 more flavors of ice cream. We've increased already the chemical diversity. We don't know much about the downstream pathways of CPS3 and 4, but we're really excited about what might lie beyond. So let's go back to ZMAN1 and AN2 and what the pathway looked like before and now. Again, we've got the gibberellins on our left, and then you can see in the center in yellow, the coralexins, and we've now filled out and elucidated that pathway, along with Eric Schmel's group at UC San Diego, who we're really happy to do a lot of collaborations with. What's interesting here is that you can see the coralexins proceed through N-isocorine, the gibberellins proceed through N-corine. In theory, the coralexins can proceed through N-corine, but it's interesting here that the ZMKS2 is providing a mechanism to separate out the growth and defense pathways, a secondary method in addition to the separation of AN1 and AN2 for different functions. Now, what is really exciting here, and is the focus of the remaining of my graduate research, is this new pathway on your right in red, the delabrolexins. You'll notice that this was not even on our original drawing because we actually found these not through metabolite to gene approach, but gene to metabolite approach. So upon transforming some genes into E. coli, we found these molecules, and I'm going to show you the pathway next, that we had not ever seen before in maize, and frankly, some of them had not seen in nature before either. So first, ZMKS4 makes delabrodine. This was actually seen first in a conifer species found in the 70s. And then cytochrome P450s decorate this with interesting oxygen groups and to make epoxydelabranol. This group now we're calling the delabrolexins. So epoxydelabranol was a totally new molecule. So far, it's unique to maize as far as we know. And to us, it's particularly interesting because that epoxide group appears to be highly stable. Epoxides are somewhat unusual, and typically when you do see them in plants especially, they're not very stable. Now, when you're looking at this, you should be asking an obvious question, which is, why have you not seen these? Why had nobody seen these in maize before? It's been a model crop for a long time. And is this just a result of your E. coli co-expression system? And that's what we were asking ourselves because we were frankly surprised to have found these. And so we then decided to go dig into the plant. And I mean that literally, we went digging into the roots because the transcriptome told us that it was likely that this pathway was expressed in roots. And these are the results. So what you're looking at is, and I'm going to show you many of these, is using GCMS to quantify the metabolites. And here we're looking at total delabrolexins. And we found that, yes, they are indeed present in maize roots. You're seeing 3 maize varieties, B73, Golden Queen, which is a sweet corn, and Mo 17, which is a field corn like B73. And you're seeing that they actually have quite varying levels. And this was also interesting to us because B73 is a maize variety for which many of us use as our model, and it's what the first genome was of. And so it's perhaps unsurprising that nobody saw these because it appears to be a low producer. So we're excited about this variation that we're seeing between varieties, and it's something we're exploring further. We were happy to see that indeed these are present in maize roots. So next, we wanted to ask ourselves, do these delabrolexin metabolites derive from ZMAN1 or ZMAN2 in the plant? As I've drawn them, they were originating from ZMAN2, but we couldn't really prove that yet because both AN1 and AN2 have the same function. Now, fortunately, maize is amazing, and there is a wealth of mutant resources. So let's take a look at those real quick. ZMAN1, which is anther ear 1, is a mutant in the ZMAN1 gene and results in this anther ear phenotype, which severely dwarfed plants morphologically is doing something very wrong that's characteristic of a lack of gibberellins. This is how we know that AN1 and AN2 are separated mostly for growth and defense. Now, let's look at the AN2 mutant now. AN2 looks totally normal under normal conditions. We don't see these morphological defects. We do know that the AN2 mutant, at the time we knew that it lacked coralexins, and this is part of why they were how they were characterized as deriving from AN2 and why they were important for the stress response. That's because while AN2 is normal under normal conditions, it is more susceptible to stress. So let's take a look at that now. So the first is fungal stress on your left. This is infection by Fusarium verticilioides. Now the wild type has some Fusarium growing, but the mutant AN2 has a lot more Fusarium growing. It's much more susceptible. Now, Fusarium verticilioides is the cause of many types of rot in the plant, most specifically ear rot, which causes total Fusarium is about 14% crop loss in the United States every year for maize. It's huge, several $billion, and is a huge contributor to mycotoxin contamination of grain. So it's a huge issue and is why people are so excited about the AN2 mutant, because we see that it's more susceptible to stress and told us that the coralexins therefore should be important in that stress response. In addition, the AN2 mutant is more susceptible to drought stress. We see reduced root and shoot growth and weights and earlier leaf curling, and it just dies sooner. It's much more susceptible. So obviously we're excited about this mutant, and this could also tell us if the delabrolexins are downstream of AN2. Indeed, it does appear to be true. So here you're seeing, again, quantified metabolites. We do see a little bit of leakage in the AN2 mutant through one of the metabolites. That's the black bar there. And we believe that's because ZMAN1 is still functioning. If there's some NCPP around, it can be used towards the delabrolexins. But predominantly, the AN2 mutant does lack delabrolexin metabolites. This also tells us that delabrolexins may be involved in the plant stress response, like we've previously seen for the coralexins. So let's dive into that a little more. And our first place to start was at abiotic stress. Here we used copper sulfate applied to roots. Again, roots is where the transcriptome told us to look first. And we see a strong induction of these metabolites, one of which I'm showing you here, in these roots under exposure to copper sulfate stress, up to the nanograms per gram of fresh weight root tissue. So this is telling us that, yes, these molecules are not only lacking in the AN2 mutant, which is more susceptible to abiotic stress, they're also induced under that stress condition. So they are important. Next, we decided to dive into fungal stress. Again, fusarium is incredibly important economically and for public health and safety because of mycotoxin contamination of grain. It typically infects ears, but it can cause root rot, stalk rot, basically can rot anything. So here we infected roots with Fusarium verticilioides, that's fungus 1, and Fusarium graminearum, that's fungus 2. And again, see a really strong induction. I'm just showing you one of the metabolites here of these metabolites in root tissue in response to this infection, similar to abiotic stress. So next we wanted to ask ourselves, okay, why are the plants making this under fungal attack? Are they antifungal metabolites? Can they inhibit the growth of these fungi? So here you can see one of my former undergraduates, Karen. We grow the fungi on a plate, then we transfer them into liquid culture in the 96-well plate you're seeing here. We use optical density in order to measure the growth of the fungal cultures in the presence and absence of different delabrolexin metabolites. Now, if you were paying attention earlier, I said E. coli was my favorite production method in order to test out these genes to figure out what it is that they do. And that's because E. coli is great for just producing large amounts of these metabolites and allowing us to purify them. We can even separate out different molecules along the pathway in a single culture to large amounts. And this is much easier actually than extracting and trying to purify these from roots. So what you're seeing here in this graph is a growth curve of Fusarium verticilioides. The black is with no molecules added except a control. And then the blue and the red is with just one of these molecules added at physiologically relevant concentrations. And indeed, we do see a significant reduction of growth. And we do know then that these molecules are antifungal. It's something we're continuing to explore as far as which fungi can they inhibit growth of and what might this mode of action be. So as a short summary, this gene to metabolite approach, where we have metabolites we didn't know about before, has provided new insight into plant metabolism, and it's shown us a brand new class of molecules, the delabrolexins, that so far are unique to maize. These molecules also appear to be a major component of both biotic and abiotic stress responses. So what is the ultimate goal? I expect most of you on this call, like me, would really like to increase crop yields while providing a sustainable future, right? Both for farmers and for our environment and for us who have to eat. So our goal is ultimately that we can understand these pathways better, and then we can implement them in breeding or engineering to make crops that are more resistant to disease. And we have a great example of that. The Wang Lab at Sichuan Agricultural University in China actually did just this. So they took one of the enzymes in the coralexin and delabrolexin pathway that I showed you previously and overexpressed it in rice. This is a function that rice did not already have. They see that this overexpression makes brand new molecules, new diterpenoids, so we can increase that number to 10,001 that we know about now. And in addition, it provides novel resistance to rice blast disease. So it's a really good indication that this knowledge is helping and we can actually make advances towards increasing crop resistance to disease. So with that, there are many people to thank. Most importantly, my advisor, Philip Zerbe. Subangela Mafu is the postdoc who first started this project and is now a professor at the University of Massachusetts Amherst. Many undergraduates who have worked on this with me, I'm so thankful for. And most of the work I showed you today was in collaboration with Eric Schmelz and Elisa Huffaker at UC San Diego. Of course, to our funding, especially the NSF and USDA, who have funded my graduate education and research. And with that, I'm going to leave you on a slide with some contact info, both for me and for the ASPB groups that I'm involved in case you want to learn more. Thank you.
thank you, Katie, for an exciting talk. It was really like two very plant science talks from the biochemistry perspective. So things are lined up really well.
Yeah.
Cool. Thank you for this. I learned a lot about these metabolites, which I also normally put on the back burner. So it's nice. It's good to see. You know, I like the connection to the fungi because I work on fungi. So it worked for me. Okay, so there's a couple of questions coming in, so we will take those one at a time. So can you apply this? Oh, I can't even pronounce those.
That's okay. Dolabrolexins.
Yeah, dolabrolexins exogenously and complements the phenotype of your SMAN2 mutant.
Yes, this is an excellent question. Thank you for asking this. I would love to do that. At this point, I can't yet produce enough. So we're producing these molecules in E. coli, and we have pretty good titers, but it is still very time consuming. So we're able to produce the amounts we need for fungal assays, but I can't yet produce enough to apply to plants. But that is a goal in the future if we can scale up some of our E. coli production.
All right, so going on a little bit from there. So are these oh God, I just call them D now. Are these secreted to the apoplast and the roots? Like, so where, like where does it go?
Excellent question. I like these questions. This is great. So I do have some evidence that I didn't show here that these are secreted into the rhizosphere. Something I did not talk about today, I have some other work that's on the rhizosphere microbiome and the involvement of these molecules and the coralexins, this other group, in shaping the maize rhizosphere microbiome. So there wasn't time to share that today, but if you go to Plant Biology 2020 Virtual Summit, which I'm very excited for, I will be giving a talk on how these are involved in the microbiome and what we know about their secretion status.
That sounds really interesting. Go get your seat at Plant Biology later this year. All right, so kind of along this lines as well. So you showed this antifungal effect of your D molecules, and so the question is, how does it actually inhibit the fungal growth, and is it also applicable to other Fusarium races, strains, like species, like how you look at this? For example, for the Fusarium R4 race, which infects banana.
Great question. So this is something that we're actively working on as soon as we can get back into the lab. So we now have a catalog of, I believe, 8 or 9 different Fusarium species, because we're interested in, is this applicable to only the ones that are pathogenic to maize or to others? So we're looking at Fusarium oxysporum and solani that both infect tomato, Fusarium subglutinans and proliferatum. We do not have the race that infects banana, and I believe we don't have a high enough BSL to test that. But we are investigating how they're affecting other Fusarium species, and I can tell you the preliminary evidence shows there is variation between the different races of Fusarium. We do not yet know the mode of action, so how they're inhibiting growth, but that's something we're actively pursuing, and we're using imaging to look at the fungi and how it compares to other fungicides if we apply those in this assay. And that's also going to give us an estimation of how good are these as pesticides, really.
Thank you. So there are 2 kind of related questions, and I packed them into one. So you do some overexpression of your biosynthetic genes. So do they have negative effects on the plants if you overexpress and presumably then also overproduce your metabolites? And related to this, how are these clusters normally regulated in the gene expression level?
Great question. So first, we do not have any overexpression lines in maize, and the lab in China has overexpressed one of the maize genes in rice. So based on their paper, they don't see any negative effects thus far, which is really interesting. We don't know yet in maize. We would love to get some overexpression lines to see, right, does this have any effect elsewhere in the plant, especially in those gibberellin pathways which share a precursor. We also don't yet know about regulations. So it's really interesting to us that you have ZMAN1 and ZMAN2, which make the same precursor but appear to funnel into these different pathways. And we think that's based on the timing of how those are regulated, but we don't yet know much about the transcription factors. It's something we are actively exploring and are excited about. But yeah, to be determined.
All right, so a couple of bunch more questions, and we will forward you to them later, and maybe some can be also answered on Twitter. I would like to ask my personal question
Yes.
at the end, which is a little bit like further off. So you see all these plants that do all these millions of metabolites. So, and there's so much specificity that each plant species, or maybe even like a single accession from a plant species, produces different metabolites. Like, why do you think that is? Why wouldn't be like 1,000 metabolites or 10,000 metabolites like used by all plants in general? And that would be perfectly fine, and you could achieve everything with just these 10,000 or 20,000 with a limited number. Why is it so diverse?
I think that's an excellent question. I think about this a lot of why do we see this and why do we see so much species specificity? And I think part of it is just the cost. I mean, you're spending 20 carbons on every single one of these molecules. And it's an incredible amount of energy to make these and to make the proteins that have to go into it. So why we don't see more defenses, I'm not really sure, but I think if you've got what you need, you're not going to make stuff that might be wasteful based on energy in your carbon sources.
Cool. Thank you very much. So thank you to Katie and also to Florian, two very exciting biochemistry talks today. I think that would spot on plant science. We will forward all the questions which were remaining. I would like to thank once again ASPB and Katie, Jürgen and Mary for a really exciting sessions over the last couple of weeks. There will be a couple more, I think 3 or 4, before the ASPB plant biology break. I would also encourage you to investigate if you like to attend this virtual conference. Sounds pretty exciting overall. So have a look, and I hand it over to Katie. Thank you.
Yes, thank you all for attending. We will be posting the recording of this webinar within a few days, so be on the lookout for that. You can find it on our website, plantae.org, under the Plantae Presents tab. Yeah, thank you all, and hope to see you next time.